In this talk about innate immunity, we're going to learn about antimicrobial molecules that are made by the host. We will learn about different antimicrobial molecules that are made by the host in response to pathogens, as well as their mode of action. The host makes many different kinds of antimicrobial molecules in order to limit pathogen growth and colonization. Some examples are Lysozyme, which is an enzyme that is found in the saliva and tears, as well as antimicrobial peptides that are made by different cell types in the host. Now, when we look at this picture, we're looking at the intestinal lining, and we can see that the intestinal cells produce and secrete antimicrobial peptides into the gut lumen. And these antimicrobial peptides can limit the growth and colonization of microorganisms in the gastrointestinal tract. Now, the host can make many different types of antimicrobial molecules, and here is a list. Now, this does not include every single antimicrobial molecule, but you get an idea of all the different types of antimicrobial molecules that are made in order to defend the host from pathogens. So let us look at some of them right now. Many metal ions like iron and calcium are essential for the growth and proliferation of any type of organism. Now, the host makes proteins like transferrin and lactoferrin, and the job of these proteins is to bind to iron so that iron is not freely available in the environment. Now, if we look at the figure here, we can see lactoferrin and transferrin, and they are able to chelate these iron ions. And so they bind to the iron ions, and thereby the iron is not freely available in the environment. Now, in the case of microorganisms, they also need iron to be able to grow well. And so some microorganisms have made molecules called siderophores. And these siderophores are able to bind to iron also, and that way they are able to store iron and get iron from the environment. When we look at this picture, here we can see the scenario where the microorganisms are making siderophores, and these siderophores are able to bind to iron. Once they're binding to iron, they're able to give the iron to the microorganism, and the microorganism is able to successfully colonize in the host. Now, there is a tug of war that happens in the host. So the host cells will try whatever they can do to limit microbial growth. And on the other hand, microbes will try whatever they can do to survive and colonize. Here's an example where the host cell makes another molecule called NGAL. And what NGAL does is that it will actually bind to siderophores that are made by microorganisms. So this way we can look at an example where the host is trying to limit an essential nutrient like iron by making proteins like lactoferrin and transferrin, while microorganisms are able to make siderophores that are also able to grab this iron. In response to the ability of microorganisms to make siderophores, the host cells can also make another molecule called NGAL that is going to bind to this siderophore. Lysozyme is an enzyme that acts as an antimicrobial molecule, and it is found in many secretions like tears, saliva, and other bodily fluids. Lysozyme acts on the peptidoglycan of bacteria. So when we look at the figure, peptidoglycan is present in both gram-positive and gram-negative bacteria. It should be noted that the layer of peptidoglycan is a lot thicker in gram-positive bacteria as compared to gram-negative bacteria. What lysozyme is able to do is it is able to cleave the glycan chains that keep the peptidoglycan intact. And so by cleaving these different bonds, we can see that the integrity of the peptidoglycan is compromised, and thus we have exposed lipid layers where other molecules can then act on the phospholipid bilayer of the bacteria. This way, lysozyme plays an important role to limit bacterial growth in the host. Secretory phospholipase A2 is a highly basic enzyme that is found in body secretions and fluids like tears and saliva. It is able to cleave phospholipids that are present on the plasma membrane, and thereby the integrity of the membrane is lost. Here is an example where we can see the action of secretory phospholipase A2 on a bacteria. Now we should remember that secretory phospholipase A2 acts on the phospholipids. So if there is a breach in the peptidoglycan cell wall, then the secretory phospholipase A2 is able to access the phospholipid cell membrane of the bacteria, and it is able to cleave the phosphate group from the lipid moiety, and thereby it compromises the plasma membrane. Lectins are a family of carbohydrate binding proteins. Some lectins are bactericidal in nature, and usually these ones are made by epithelial cells. A good example of a lectin that is antimicrobial in nature is the Reg3 family of proteins. These proteins are made in intestinal cells and are secreted in the lumen, where they can act on microorganisms that are in the lumen of the gut. Reg3 is activated by trypsin, which is an enzyme that is found in the gut and helps in digestion. Activated Reg3 is able to bind to peptidoglycan and can form pores on the bacterial surface, which is what can be seen in the figure to the right where the orange circles are. Antimicrobial peptides are a class of peptides that are an ancient form of defense against pathogens. They are secreted by epithelial cells as well as some immune cells which act as phagocytes. The epithelial cells secrete them into the surrounding fluid, while the phagocytes are able to secrete them into surrounding tissues. The three main antimicrobial peptides that we are going to look at are the defensins, the Cathelicidins, and the Histatins. Let us first look at the defensins. The defensins are antimicrobial peptides that are found in many eukaryotic organisms, and that is why it is considered as an ancient form of defense. It is a peptide, and hence it's approximately 30 to 40 amino acids long, and it forms an amphipathic structure. They're normally synthesized as propeptides that have to be activated, and the activation occurs through proteolytic cleavage. In this figure, we're able to observe where the cleavage happens and the amphipathic nature of the antimicrobial peptide. There are three subfamilies of defensins that are found in organisms. The alpha defensins are normally made by neutrophils and Paneth cells. The beta defenses, on the other hand, are made in response to microbial products, and they're usually synthesized by the epithelia that are outside the gut. The theta defensins are not made in humans but can be found in other organisms. They are not made in humans due to a mutation that has inactivated the gene. When we look at the mode of action of defensins, their main mode of action is the disruption of the cell membrane. They're able to act on various bacteria and fungi, and the way they're able to disrupt the cell membranes is that they insert their hydrophobic regions. So when we look at the picture, here we can see the defensin, and we can observe the yellow part, which is hydrophilic in nature, and the green part, which is hydrophobic in nature. So they insert their hydrophobic regions in the cell membranes, and they're able to disrupt the membrane bilayer by forming a pore. Due to this pore formation, the cell membrane becomes leaky and the contents of the cell leak out, while other things in the environment are able to enter the cell, and this is how the cell dies. Cathelicidins are antimicrobial peptides that are constitutively made by neutrophils and macrophages. They are similar to defensins in that they are synthesized as propeptides that are inactive and can be activated by proteolytic cleavage. They are made in response to infection by keratinocytes in the skin, as well as epithelial cells in the intestine and lungs. Thereby, depending on the cell type, either they're constitutively made or their synthesis is induced. Now, when we look at the mode of action of cathelicidins, here we can observe the epithelial cells making the cathelicidins, and these cathelicidins are activated through proteolytic cleavage. And when they're active, what they're able to do is they bind and coat the cell membrane. This coating of the cell membrane results in the membrane dissolution because the membrane starts forming micelles. Due to this, pores are formed in the plasma membrane and the intracellular contents are leaked out into the environment. And this way, the pathogen is killed. Histatins are another group of antimicrobial peptides that are made in the oral cavity by salivary glands. They are short histidine-rich cationic peptides and hence the name histatin. They usually act on pathogenic fungi and promote wound healing in the host. So the way they're able to act on pathogenic fungi is, here we can see a histatin, and it first attaches to the cell membrane of the fungus, and once it enters the cell, it targets the mitochondria. Now, the mitochondria is the part of the cell that synthesizes ATP, and it makes the mitochondria leaky, due to which ATP is released from the mitochondria. This way, the mitochondria loses ATP, and this ATP loss can activate cell death pathways. Now, in addition, that loss of ATP also restricts cell respiration, and thus metabolic pathways are also impacted. It also results in the formation of reactive oxygen species, or ROSs, and these reactive oxygen species induce G1 cell cycle arrest. So this way, the fungus is unable to survive properly, and the histatins thereby are antimicrobial in nature. The complement proteins are a collection of more than 30 different soluble proteins that are present in blood as well as bodily fluids. They were initially discovered in vertebrates. However, now we know that homologs of some complement proteins are also found in invertebrate organisms. A lot of complement proteins are thioester proteins, and when we look at vertebrates, the complement proteins are mainly synthesized in the liver. Now, the complement proteins work together and form a cascade that's called as the complement pathway. The complement pathway aids in the pathogen elimination as well as induction of inflammation. These complement proteins on their own are not antimicrobial in nature, but when the complement pathway gets activated, that is when pathogens can get eliminated. To summarize the different barriers that are present on epithelial surface that protect the host from different pathogens, we have mechanical barriers as well as chemical barriers. These chemical barriers are in the form of many different types of antimicrobial molecules that include acids that generate a low pH, like hydrochloric acid that is found in the stomach, as well as other enzymes like Lysozyme or Secretory phospholipase A2, and also antimicrobial peptides like defensins, Cathelicidins, and Histatins. There are other types of chemical molecules that are made that are antimicrobial in nature. And we also have microbiological barriers in the form of the normal microbiota that is present in different epithelial surfaces of the body. With this, we come to the end of the talk where we learned about different antimicrobial molecules that are synthesized by host cells as well as their mode of action.